Dear students, today we will be dealing with the module Environmental Biotechnology. In that, we will be specifically dealing with what are the methods that are used for detection of pollution using biotechnological methods. So that will be the first topic we will be covering in this module. So what is environmental biotechnology? How can we define environmental biotechnology? It's simple. It's actually the application of biotechnological principles. You know that there are several principles involved in biotechnology, and which is mostly related with biological organisms, using of biological organisms in several technological processes. So we will be applying the basic principles of biotechnology to solve problems associated with the environment. The problems associated with the environment, we mainly deal with pollution. Okay, so environmental biotechnology, in principle, is the application of biotechnology in problems associated with the environment. Environments means maybe natural environment, agricultural, or man-made environment. And the major aspects of biotechnology, environmental biotechnology, are bioremediation. We'll go to detail each one of this. Bioremediation, waste management, bioleaching, then using development and using of biopesticides, biofertilizers, bioleachers, etc. And mm, actually another aspect of this environmental biotechnology is using microbes, microorganisms. You know that they are invisible forms. That many of them are yet to be identified. Okay, and they mostly involve bacteria and viruses. And there's one term you need to know, that is metagenomics. Metagenomics means the analysis of genomes of the entire communities of microbes. Okay, so here in metagenomics, we analyze the genomes of the entire communities of microbes instead of analyzing each one of them. Understand? So the analysis of genomes of entire communities of microbes bypasses the need to isolate and culture individual species. We need not do the genomic analysis of individual microbial species, but together we can do the microbes as a community. Okay, and this metagenomics is based on the principle that nucleic acids, based on principle that all creatures contain nucleic acids, and these acids encode various proteins, and therefore all organisms need not have to be cultured, but they can be identified. Each one of them can be identified by a particular gene sequence or protein or metabolic incorporate. So that is what is metagenomic. So that's the analysis of the complete genome of the complete microbial community. And let us see what are the biotechnological methods used for pollution. And we know that environmental pollution poses a big threat to the existence of humankind. And to deal with pollution or to mitigate the pollution, we need to know how much polluted it is. Each environment, we need to know how much polluted it is. Then only we can design measures to mitigate that pollution. Okay, and how these biotechnology methods are used for detection and also monitoring of environmental pollution is using the biosystems. What are biosystems? Biosystems is model biological organisms which can be used to detect and abate pollution. Okay, and in these several groups of plants, animals, and microorganisms are used. And when we choose these biosystems or biological organisms to deal with pollution, either to detect it or to monitor it or to mitigate it, we need to these bioorganisms should possess certain parameters. They should be reliable. Okay, reproducible. When I use a bioorganism in this India, it should be applicable in America also. Okay, there is reproducibility. One experiment we do here should be reproduced somewhere else, and it's also should be cost effective. And usually three major types of criteria are used for biomonitoring of pollution, and there one is visual rating, another one is genotoxicity rating, and third one is metabolic rating. Visual rating, genotoxicity rating, and metabolic rating. And visual rating, what visual rating? See, we are assessing visually or seeing it, what are changes take place. Okay, so this is the easiest and cheapest method of biomonitoring, and here we monitor the growth rate and productivity of the organisms we use for monitoring. Suppose we use a particular bacterium or use a particular algae, we visualize or we see or monitor how much growth rate it has affected, how much productivity it has affected, whether that is due to the pollution, etc. Okay, and for example, if you use microorganisms, use carbometric analysis. That means microorganisms produce turbidity. When they grow, they produce turbidity. So the amount of turbidity, we can use it as a measure of how much pollution it may take place. Okay. So turbidometric analysis, and if you use higher plants, you can see the growth rate, whether it growth rate is up or down. Okay, so growth rate of different parts of the plants, how much damage it causes to the leaves, how the seeds are viable, is it less viable or more viable, and germination frequency, etc., can be used as measures for detecting the pollution when we use higher plants. Okay, and when we use animals like fishes, the toxicity levels are okay, for example, you might have heard about LD50. LD50 is lethal dose 50. And lethality, it is causing death. And LD50 means that how much concentration of this pollutant is needed for the mortality of 50 percentage of that particular organism. Okay, for example, if we use a fish like tilapia for a pollution detection, and we use 100 tilapia seedlings, we measure at each concentration of that pollutant how much or how much concentration of this pollutant is required for 50%. That is 50 fishes should be to attain the mortality of 50 fishes. Okay, that is lethal dose 50. So that fishes can be used for pollution, and sometimes either simply presence or absence of a particular species of organism can be an indicator of pollution. Okay, we know that bioindicator organisms. We know that some frogs, amphibians, amphibians can breed in almost pure water only. Okay, so that means that if amphibians are present in a particular water body, that water body is comparatively purer, okay, or less polluted. Okay, so these are just the presence or absence of a particular species of organisms serves as an indicator. Okay, so these are some of the bioindicator species, some fishes, frogs, then some plants and some earthworms. These are all bioindicator organisms. So that's about visual rate. And second one is genotoxicity rate. What is genotoxicity? Genotoxicity is the damage or deleterious action that happens on the cell's genetic material, that's the DNA or nucleic acids. Okay, so nucleic acids, if they get injured or some deleterious action takes place, the whole integrity of the organism is lost. Okay, so here many pollutants have a genotoxic effect, and these injuries or damages that cause in the genetic material is called mutation. Okay, so the pollutants have the capacity to produce mutations in what this genetic materials. And there are several genotoxicity tests which are used for detecting the presence of such pollutants. Okay, and we can test. These tests can measure how much extent this damage has taken place, either it's at cellular levels or subcellular levels, how deep it goes. Okay, this can be tested using genotoxicity rating. And there are several lesions. Lesions means small injuries. That might be also problems. And these genotoxic lesions can be detected on cellular organelles like plasma membrane, mitochondria, etc., and also in genomes, immune systems, biomolecules. And there are several cytotoxic tests which can measure how much amount of damage has taken place in chromosomes, then chromosomal exchange, and also for pollution. And another one is cell viability. How much viable the cell is can be detected using in vitro lysosomal viability. You know that lysosomes are cell bodies which contain enzymes. Okay, and these are the bodies which can collect and collect and kill the toxic substances that are produced in our body. So cell viability can be measured by how much effective the lysosomes are using in vitro studies. Okay, DNA probes are recently also used for detecting the pollution in water. So that is VISO, visual rating, and genotoxicity. Next is metabolic rate. So you know that all these changes which take place either in morphology or in genetic material will cause biochemical changes in the body. Okay, body of the organisms. And in metabolic rating, we measure what all metabolic changes takes place in the selected organisms used for detecting the pollution. Okay, that may be qualitative, just qualitative measuring, or we can measure it in numbers. Quantitatively, we can measure. And certain metabolic parameters used as biomarkers. It can be used as biomarkers for assessing how much stress the organism is due to pollution. And these biomarkers include chlorophyll, the amount of chlorophyll present, then proteins, nucleic acids, and also change in the enzyme activity. That's what is called metabolic change. So these enzyme activities will get changed after pollution. Okay, so we can assess how much changes have take place due to metabolic, this pollution. And then we come to bioassay, or measuring of how much pollution has taken place using whole organisms. Okay, then here the bioassays are conducted to measure infectious substances on a living organisms as such. Okay, so when we choose an organism for bioassays, it is expected to satisfy some criteria. Okay, and when we use an algae or a plant or a fish or whatever it is, when we use it as a bioassay marker, it should readily take up the pollutant. That means it should be sensitive to pollutant. Okay, it should be able to take up the pollution if it is in water or soil or air or whatever, wherever it is. It should this organism should take up the pollutant, and it should be sensitive. It should not be resistant to the pollutant, and should possess measurable features. Okay, and we know that this particular fish can take up this particular pollutant, and that all theory we know that. But if we don't know how to measure that pollutant, how much amount of pollutants it can take place, then there is a no use. So this organism should possess measurable features to detect pollution, either visually or genotoxically or metabolically. Okay, and the organism should have wide occurrence. It should have wide occurrence. That means then this experiment can be repeated in anywhere else, at least in the same locality, and this organism should be available around the year. Okay, should not be seasonally available. And this bioassay should be simple, reproducible, and cost effective. And now see what all organisms, common organisms use for whole organism bioassay. And we can use algae, bacteria, lichens, mosses, then add some animals, macrophytes, all for bioassays. And algae that we commonly use include chlorella, microsis, spirulina. These are all navicula, then fucus. These are all common algae which are used for bioassays of pollution. And coming to bacterial bioassays, and usually bacterial bioassays are used for water pollution analysis. Usually fecal pollution. Okay, pollution caused due to fecal emission or fecal contamination. And the most common test we used for fecal contamination test or bacterial bioassay is coliform test. That means we use coliform test means we use E. coli. And if you find at least a trace of E. coli in drinking water, potable water, then that water is not potable. This E. coli can come to water, drinking water, only through one source, that is fecal contamination. So even if one E. coli is present in that drinking water, and that is not usable. So that can be tested using coliform. We know that some public laboratories can test our wells and bore wells, etc. How much pure the water we have. So what we do primarily is coliform test. Is this water drinkable? Is this water potable? And they test using coliform test. And also bacteria salmonella. NAMES test can be used for detecting mutagenic pollutants. And there's another thing called bacterial bioluminescence. Okay, here bioluminescence, this what lighting up. Okay, then is another technique used for the measurement of gaseous pollutants and other compounds. Example: sulfur dioxide, formaldehyde, ethyl acetate. These all can be detected by bacterial bioluminescence. And the common bacteria used for this purpose is Photobacteria fosforia. Okay, and then coming to lichens, atmospheric gas pollution. For detecting the atmospheric gas pollution, lichens can be used because they are very sensitive to sulfur dioxide. You know that sulfur dioxide is a very common atmospheric pollutant, and this can be detected by using lichens. And Moses also can be used for detecting the environmental metal pollution. And example, stereofilm, speculum, brightness, these are all Moses used for detecting metal pollution. And then vascular microfilms. We know that in our water bodies, there are so much of vascular microphytes like water hyacinth, duckweeds, then we call as African pile, etc. Okay, so aquatic metal pollution can be detected using this vascular microphytes like water hyacinth and duckweed, and several animals are used for pollution, mainly fishes and helminths. Okay, fishes are very commonly used for bioassays of polluted and aquatic habitat pollutions, and mm, the critical parameters used for the assessment of bioassays include the changes in morphology of fishes. How much, what all changes take place in the morphology of the fishes due to pollution. We use it as a model organism of pollution. Okay. And also the behavioral patterns, the gill movement, the movement of the operculum. These are all visible changes. We can detect the effect of for the presence of pollution. Okay. Then alteration in enzyme activities also serve as a reliable marker for pesticide pollution. For example, acetylcholine esterase. And the main fishes used for the common fishes used for pollution detection are cutla, labio, chenna. These are all fishes with a very hardy skin. Okay, so they can be very commonly or very easily used for detection of pollution. And some we saw different types, algae, lichens, mosons, animals, fishes, etc. We saw, but it's very good to use a multiple species of organisms together because we know that lichens may use atmospheric gas pollution. At the same time, vascular microphytes are used can detect metal pollution. Okay, so instead of using singular organisms for bioassays, it's good to use multiple species of organisms for pollution detection. And then coming to cytogenetic bioassays, they are often exploited for environmental pollution detection with reference to mutagens and carcinogens. Carcinogenic pollutants can be assayed using cytogenetic bioassays, and here the harmful effect of pollutants on different cellular components like chloroplast, mitochondria, chromosomes, etc., can be detected using cytogenetic bioassays. Okay, and here macromolecules like nucleic acids, proteins are also used for detection. And there is one test called micronucleus. Here, um, micronuclei are found after the fragmentation of chromosomes. And a small, this what cytoplasm will cover and small amount of micronuclei will form. And these micronuclei can be used for screening of the mutagenic compounds. And there are several molecular probes and immunoassays available for pollution detection, and these are actually molecular probes useful for detection of bacteria, virus, and other pathogenic organisms that cause diseases. And immunological techniques like are also used for detection of pollutants, identification of pathogens that causes immunological. Okay, and these immunoassays are also used for detecting several pesticides like DDT, thiazines, glyphosate, etc. And these are all modern techniques. And also biometabolic products of certain bacteria also can be used using immunoassays. And for example, the toxins produced by bacteria like chlorella and salmonella, these are all can be detected using specifically designed metabolic immunoassays. And there are biosensors available. So biosensors means when we use certain organisms for pollution detection, it may take some time, its own time for developing the symptoms on the organisms that we use or the visible changes. So it will take some a certain time. But when using biosensors, that is technology associated with this principle. Okay, and get a more quicker okay, so biosensor is an analytical device containing an immobilized biological material. So biological material is there, okay, but technology is more used. So like enzymes, organelles, or cells. this type of biological material will be there, which can specifically interact with the analyte. That means the pollutant, for example, the concentration of pollution or pollutant we need to be determined. Okay, so the biological material is there, and also that can interact with the this compound and produce a physical, chemical, or electrical signal. Okay, in earlier cases we see only the physical changes, but here we see physical, chemical, and also electrical signals can be detected. So we can measure quantitatively how much effect or how much pollutant is there in that particular state. And they are highly specific, they are highly accurate, and can be used for detection of environmental pollutions. The most important or most commonly used uh, example for biosensors is by BOD biosensor. BOD is biological oxygen demand. Biological oxygen demand, or BOD, assesses how much pollute, how much pollutants are there in water, or we can easily detect aquatic pollution using biological oxygen demand. And we are using, we know that planktons and microorganisms are there in water, and they use oxygen, or phytoplanktons release oxygen. Okay, so how much oxygen is required to run this system? That is biological oxygen demand. It is widely used for detection of organic pollution, and usually this BOD test, or biological oxygen demand test, takes 5 to 6 days of incubation of water, and that takes almost a week to get the result. But here, when we use a biosensor, we said that conventional BOD test gives about 5 days of incubation of water, and using a BOD biosensor, using the yeast, Cyclosporon cutaneum, with the oxygen probe, takes only 15 minutes for detecting the organic. What we get instead of 1 week, we get the result in 15 minutes. So that's the use of biosensors. That's technology integrated with a biological system. So these are all the ways for uh, detecting pollution in environmental systems. So I think you understand this topic, and if any doubt you have, please contact me. Thank you.